Everybody, my name is Gerd Michel. I'm the Chief Scientific Officer of Vela. I'm based in Singapore, whereas our speakers today are all coming from Europe. And I will give you a brief introduction into the Vela systems and show you some data on HIV and HCV resistance and genotyping testing. Before I go ahead, may I ask the audience, how many of you have attended our workshop last year at the ESCV meeting? One. Oh, great. Okay. I'm asking that question. And the other question which I have is, who attended Professor Perno's lecture this morning on HCV and DAAs? Okay, so the majority, then I don't have to go into detail with this one. All right. We have developed a workflow for next-generation sequencing based on ion Torrent over the last couple of years. And this is our technology concept, which is shown here. The workflow starts with automated sample preparation for DNA or RNA, and then the user makes a decision on either going into NGS or into qPCR assays. At the end of the workflow, either PCR or NGS, we have a Sentosa link system which combines the data and interprets the data and can also be connected to the laboratory information system. So it's one workflow for HCV and a different one for HIV. However, the time, as you will see, and the hands-on time and the total assay time are very similar. Now, as I said, our technology is based on ion Torrent sequencing. Most of you may have heard about it. I don't have the time to go into a lot of detail on the technology, but it's sufficient to say we're using the so-called 318 chip that has been developed by Thermo Fisher a couple of years ago. And what we're measuring is pH changes upon association of DNA bases, which gives translation into electrical signal which is recorded. The work shift analysis, the workflow analysis for all these, for both HCV and HIV is shown here. We have a start with, as I mentioned, a robotic system here, which was co-developed with Eppendorf in Germany. We're generating amplicons on the Verity cycler, and that amplicons, depending on HIV or HCV, of course, they're different. They're going into the library preparation, which is then followed by overnight incubation and sequencing, and final data analysis. The data analysis gives you two different detailed reports. One is a quality report that gives you information about the quality of your specific NGS run, and the other one is the clinical report that gives you information about the case you're investigating. So what's also shown on this slide is hands-on time, indicated here. The overall hands-on time on the whole workflow, it depends on the lab, of course, but it ranges between 2 and 2.5 hours. The overall assay time ranges between 2 and 2.5 days. So it's mostly automated, very little hands-on time, and you get a full report in the end. Talking about HIV, which is our major topic today, I'll give you a little bit of introduction to HIV. I won't have the time to go into all details, but I'll also touch on HCV in the end, which is not necessarily the topic of the major topic of the meeting, but depending on, or in view of the talk by Professor Perno this morning, I thought it would be interesting to give you at least some information about the HCV genotyping assay as well. So this is what we do in HIV genotyping, sequencing. We are generating two amplicons, 1,000 and 1,500 base pairs, and we're concentrating on the protease RT genes and the integrase genes. So within one run, you're getting data for all the three genes in terms of drug resistance. Sorry. So this is a technical summary of the assay characteristics. The first items I already mentioned. The throughput is 16 samples per run, 15 clinical samples and one control. We're using two different controls. One is a run control, external control, and every single sample has also an internal control. So you would know if there's any inhibition or any failure on the run. We published that design control, control concept in the Journal of Applied Laboratory Medicine last year. It's a comprehensive report which is available from Vela. The coverage, the automated reporting, as I mentioned, is done on the software developed by us. It's a so-called Sentosa reporter. And the sensitivity for the assays is about 1,000 copies. 1,000 copies means that this is a statistical value. As you may see in the following slides, but also for our speakers today, we can detect well below 1,000, but the 1,000 is a statistical threshold where we guarantee 95%. Now, when you have HIV drug resistance monitoring, of course, all of us know that there is one reference database which has been the #1 in the field, which is the Stanford database, established by Professor Bob Schaeffer in the early 2000s. And we're working with the Stanford team, as I will show you in a second, in terms of doing joint studies, but also we took a license for this database. So we are legally able and allowed to report, according to the database, our results in terms of a printout or a report. Working with the Stanford team in particular, Bob Schaeffer's group and Ben Pinsky's group this year in May at the American Clinical Virology Conference presented data which are summarized here. It was a blinded comparison of an in vitro of our NGS assay with the Sanger sequencing, which is being used at Stanford as a reference method. They looked at 49 cryopreserved samples, mainly HIV subtype B, because of the high prevalence in the United States, and there was also one subtype G. And it was, as I said, it was a blinded comparison. So what they did, they sent the sample blinded to Vela laboratories for testing. We generated the data and sent them back to Stanford. And the data were analyzed at Stanford by themselves. The focus was on RT and protease genes, and there is another study going on right now on the integrase gene. The selected samples that had large numbers of the most commonly occurring clinically relevant NRTIs, NNRTIs, and PIs as drug resistance mutations. Now, these are the one slide for the findings. They found that 3.9 mutations were detected solely by NGS, and 0.5 mutations were detected solely by Sanger. Also, 0.8 DRMs were detected solely by NGS, and 0.1 DRMs were detected solely by Sanger. And the NGS detected, as you may expect, because of the higher sensitivity, an increased number of mixed bases per sequence. And in addition to this, they looked at the concordance between the two methods, and they found 96.1%, and 3.9% of the data were discordant. If they look at the discordant data, for the 18 discordances, NGS was more resistant than Sanger, so indicated higher resistance than the Sanger assay, and Sanger was indicated more resistance in one case. So overall, the NGS seems to be reporting higher levels of resistance, at least in that system. These are the conclusions of the study. The overall concordance, as I mentioned, was very high. Sanger usually detects nucleotides variants present at levels of about 20 or higher. As we all know, those of you who are doing Sanger sequencing, is not the most sensitive method. With the Sentosa system, you're getting down to 5%, increasing sensitivity, and thereby also detecting a higher number of minority mutations. NGS assay appeared also more sensitive for the detection of low abundance amino acid variants, as I mentioned, and it showed a higher predicted level of drug resistance in 18% and in 3.7% of 490 drug resistance interpretations at Stanford, which the most commonly affected RRV class being the NNRTIs, which is also not too surprising, as we would expect. So that study, in addition to the other studies we'll see today and which have been published elsewhere, is giving quite a good picture about the performance of this HIV drug resistance assay. Another study which has been going on for 3 years now in Thailand, this is the Thai Reference Center for HIV, and this laboratory is using the assay on a routine basis since the beginning when we introduced it. And they have tested, this is data from last year, they're published in Clinical Chemistry, and as of today, they have tested more than 5,000 patients on this assay. As you may know, in this area, Southeast Asia, we have a high prevalence of the CRF1 HIV variant, which is not as frequently seen in Europe and the United States. So, whereas the Stanford data is mainly based on subtype B, this data is mainly based on CRF1. And it's showing equivalent or very, very similar concordance between the Sanger assay, between the, in their case, the Siemens TruGene assay, which is now taken off the market, and our assay. And also the QCMD testing was completed with 100% accuracy. Now, on HCV, very briefly, this is the genome of HCV, which is schematically depicting the 3 areas we're doing the sequencing on, NS3, NS5A, and NS5B. And this is data from the laboratory at the University Hospital of Singapore, which, who have tested the prototype of the assay at the time, also published in Hepatology in 2015. I'm bringing up that slide, which I've shown sometimes before, to show you that the line probe assay, which has been used most widely worldwide, has quite a significant tendency to wrong calling of major genotypes and subtypes. I'm not blaming anybody. It's been around since 1992, I think, but it's been indicated also today in Professor Perno's lecture this morning that genotyping is of utmost importance, and I will show you in a second why. There are several reasons for this. I will show you only one, which is mainly cost, and that is because Sanger sequencing and our sequencing assays are calling these genotypes very, very accurately. In addition to this, with these NGS assays, you get very accurate subtype detection. You know, there's more than 70 subtypes in HCV described in the literature. And the subtypes are becoming more important. As we know, H1A and 1B has been on the agenda for a long time. But now we're also seeing other subtypes emerging, like the 4R, which has been presented this morning in the lecture, which was very difficult to treat. So we're far away from the situation that pharma companies try to tell us that the pan-genotypic DAAs, which are used widely worldwide, are really equally effective in all the different genotypes. That's probably not true. And that came out clearly this morning in the lecture as well. We've been asked a number of times about recombinant HCV. And it's been obvious for a long time that HCV doesn't really generate these number of recombinants as we see it for HIV. However, there are a couple of recombinants described in the literature, as shown here. They are at least up to the publication date by Jens Buch's paper in 2014. There were 8 intergenotypic recombinants described and 9 intragenotypic recombinants. However, if you look at that picture, the only one which is really clinically relevant is the so-called St. Petersburg variant. This has been described by a group from Karolinska Institute in 2002 already, and this has been spreading from Georgia several decades ago into Russia by migrant workers and has been discovered in St. Petersburg, now moving towards the west, towards Central Europe. So like in Germany, they're seeing more and more of these recombinants. Why is that important? If you look at the similarity plot and the bootstrap analysis shown here, you can see that the breakpoint for these recombinants is in NS2. If you use the line probe assay for genotyping, you get a 1B result, right? Because the genotyping for this assay is done in your 5' UTR region. If you do this genotyping in NS5B region, which we are doing exclusively, you get the correct result in terms of treatment. Why? Because the NS2 breakpoint is upstream from the treatment in NS3A, in NS3, NS5A, and NS5B. So with that assay, with the NS5B sequencing, genotyping, you get the correct answer to do the right treatment. you're getting a wrong information if you're using the line probe assay. In addition to the fairly high failure rate of the line probe in several laboratories. Now, another reason why this is important, there are new drugs coming out, as you know, DAAs. And one of the drugs which came out, this was approved in 2016, end of last year, is a Sapetier by American drug company. And that has been demanded by the FDA that in the package insert, they indicate that NS5A resistance testing should be mandatory. Why? Because if there is a mutation in the NS5B for resistant mutation in the NS5A, then the treatment time will be extended from 12 to 16 weeks. And that has financial impacts, as shown here. If you have a wrong genotype, thereby a wrong treatment, selected a wrong treatment regimen, then the patient will not be monitored usually during the therapy, but only at the start and at the end of the therapy. Apart from the fact that there is a high number of relapses, as Professor Perner showed this morning, the cost will be tremendous, as you can see here. This is US data, but I think in Europe the cost is very similar. So you're wasting a lot of money by inaccurately determining the genotype or the subtype. In addition to this, you want to know if there is a resistant-associated substitution before the treatment at baseline. With this, I'll stop here and take over to Dr. Raymond from Toulouse, and she'll tell you about their experience with the HIV drug resistance assay. Thank you very much.